Good morning.
Good
Good
morning,
morning,
sir.
sir.
Yes. Fine. So for today's record, we have only 15 students. 10 of you are missing. Don't know what's the problem. Let it be. So let's start. I think Akshay is not able to give the presentation. He has given some reason. So maybe I'll start with the part, or I'll continue with the part which I have stopped at. Discussing about the tertiary structure of the protein. So we'll be continuing with that particular part only. So just for a revision, we have seen that a given amino acid may interact with another amino acid and it will form a bond called as an peptide bond. Now this peptide bond, it initiates the folding of the protein. Okay, the moment the peptide bond is formed, the protein folding process it will take place. So as your amino acids are joined to each other, it may form a polypeptide. So simply that sequence of the chain, we refer it as what? A primary structure. But whenever this primary structure is being synthesized, at the same time, there is something called as an local conformational which are taking places. And these local conformational changes which are taking place, they lead to the secondary structures. Okay, and those prominent secondary structures which are found into the protein, we have seen that they are of two categories. Either it could be an alpha helical structure or it could be a beta sheet structure, right? So these two prominent structures are found. Now, after formation of this secondary structure, as we have seen, that these secondary structures will then again interact with each other. Okay, so they will come together, they will interact with each other, or they will be arranged in such a way that they may lead to formation of the super secondary structures. And it could be the formation of the motif first, whereby these motif will again interact and they will be forming the domains. Now these motifs or the domains, what we have seen, they may be having the structural role or they may have the functional role, right? So based on that, you can now imagine that a protein could be made up of the secondary structures, that is the alpha helical or the beta sheet structures, or it may be containing some motifs, or it may be containing something, the domains, right? Now when it comes to an fibrous protein, we have seen that simply it could be made up of, or it could be dominated by the alpha helical or the beta sheet structure. It's very simple, it's linear. Fibrous, if it is fibrous, means it is linear. So the folding of that particular protein, or preparing that particular type of the protein, is not difficult. But when it comes to a globular protein, where majority of the proteins what we have, they are globular in nature. So for that, what is required, that this polypeptide chain, it should turn, it should bend many a times, so that it acquires a globular structure, right? And for that, we have seen that a given polypeptide can be having some minor structures. Okay, and these minor structures, they are nothing but the turns, the loops, the bulges. So these turns, loops, or the bulges, they help to reorient the direction of this polypeptide. Okay, or this help in twisting or bending of the polypeptide, so that a globular protein can be prepared, right? So these were the things what we have talked about. So maybe I'll continue with this, that what actually happens into the globular protein, that how this globular protein, they are acquiring a three-dimensional structure. So before actually talking about that particular thing, we'll take some examples just to understand that how these fibrous proteins, they are existing into the nature. right? So again, for revision, can you tell me the characteristic features of the alpha helical structure with respect to all the dimensions which we have talked about, just for a revision? Patkan sanga, kon sangu shakil mala, that how that helical structure is formed, how many amino acids are there per turn, the distance between them, the pitch, etc., etc. Pat sanga, koni sangu shakil? Yes, Sakshi.
Alpha helix, mm, its structure forms when the polypeptide chain twists itself into a helical conformation,
Yeah.
and about 3.6 amino acid residues per turn of the helix.
Okay. What is the distance between two amino acids?
1.5, 1.5 angstrom.
Okay, good. So, knowing this, that it consists of 3.6 amino acid per turn, the distance between two amino acids is 1.5 angstrom. So, you can imagine that it is forming an hollow rod. So, what is the diameter of that particular rod? The pitch, what we talk about. So, it is 3.6 into 1.5. So, I think so it was 5.4 angstrom. So, the pitch of that particular alpha helical structure is 0.54 nanometer. Fine. And when it comes to the sheet structure, it is it's a planar type of an structure. Okay. Where the side chains could be above or below the plane, but it's a linear type of a structure. Fine. uh, there is some reason why I asked you, because we'll require it in one of the example. Now, to talk about the fibrous protein, or to talk about the proteins which end up at the secondary structure, we'll have some examples first. Okay, first we'll understand those examples and then we'll move to the globular proteins again. So for that, let me present the screen. uh, okay, can you see the screen?
Yes, sir.
Okay. Okay, so what does this slide tell you? Means this is just the revision of the previous lectures what we have taken. So what does it say? That folding of a single polypeptide chain in the three-dimensional space is referred to as its tertiary structure. So I told you that for a given protein, whatever may be the size, it could be made up of a single polypeptide only. Within that single polypeptide, maybe the initial amino acid sequence may lead to an alpha helical structure. Then there could be a break so that it can form a turns and loops. Again, it can be a sheet, again a break, again helical, again break, sheet, something like this you can imagine. So this single polypeptide chain, it is folded many a times. It is twisted in many a times so that you get a three-dimensional structure to your protein. And that three-dimensional structure, which is stabilized by the non-covalent interactions, is referred to as what? The tertiary structure of the protein. So this protein folding, as we have seen, that the information needed to fold that particular protein, it is contained into what? The primary structure of the protein. Because the sequence is going to decide that what are the local conformational changes, what kind of amino acids it will be forming, what kind of subunits, domains it will be forming, and what will be the overall structure of the protein. So this whole information is stored into what? The primary structure. Not stored, but it is given by the sequence of the primary structure. So there you can imagine now that first the secondary structures are formed. So it could be a helix, it could be a sheets, which are forming the hydrogen bonds, or which are stabilized by the hydrogen bonds. Then further we have seen that this secondary structure, they associate, they pack close together so that they can form the, excuse me, the motifs or the domains. And the third, what you can say here, that these peptides, they tend to be short and directed. Okay, and why they are directed? Because the hydrogen bond which is forming, that is directional. Okay, and in a given protein, it may not include many complicated twists and the knots. Like suppose if I given you a thread, if you are flying a kite, you have that manja. So if you have opened up too much of that particular thread and you want to now make it into a, you have to arrange it properly. So what you do, you simply take a particular stick, and onto that you go on uh, revolving that particular thread so that it gets tied there. So there you are having too many twists or the knots which are taking place. But whenever a biological protein is being folded, it will not involve that many types of, that many times of the knots, or it will not be involving some complicated type of the twist or the knots. Fine. So even if there is a twisting or the folding which is taking place, they are the very simple type of the uh, turns or twist or the loops which are present, so that you get that globular protein. Okay, so due to this particular restrictions, ओके, आता रेस्ट्रिक्शन इथं मला काय म्हणायचंय की बघा, आपण काय बघितलं की सेकंडरी स्ट्रक्चर मध्ये फक्त दोनच प्रकार फॉर्म होतात. So just imagine that how meticulously they might have used these different 20 amino acids so that they can form only the helical structure or the sheet structure. Second restriction could be that it is not involving many turns, many loops, many complicated type of the knots, etc., etc. So they have tried to make it very simple. And due to that simplicity into the protein folding, you can imagine that these protein chains, they are arranged in few patterns. Okay, and based on those particular patterns, we have classified these proteins. So we have made the classification in terms of the families. So you have the protein families. If you go in the further studies, you may come across a protein family. So under that protein family, you may have many similar type of the proteins which are included. Fine. And these few patterns with a typical sequence of the amino acids, it has really helped us to understand the evolutionary nature or the function of that particular protein. Fine. So knowing this sequence, you can compare between the proteins. You can try to understand the evolutionary nature of the protein. The structural and functional relationship can be easily obtained from that particular sequence. This protein which is folded, the final protein, as I told you, it is a stable protein. Why it is a stable protein? Because it has maximized the non-covalent interactions. Now we have seen the different type of the non-covalent interactions. So majority of it, you can say it could be the hydrogen bond. So if the proteins, if the region of the polypeptide is onto the outer side, we can say that it is stabilized by the hydrogen bonds. And the interior, where majority of the hydrophobic interactions which are away from the water, they have come together. So they might be involved in formation of the hydrophobic interactions, the planar interactions, the van der Waal interactions, etc., etc. Fine. So with all these different type of the interactions, the protein which is being folded is a stable structure. Okay, so this is what we have talked up till now. Now we'll go to the examples. So first we start with an fibrous protein. So when you talk about the fibrous protein, the one protein which comes into our mind is the keratin. So it is one of the first protein whose structure has been elucidated, okay, or whose structure is known. Okay, we know the sequence of that particular protein, we know how it is being folded, etc., etc. So all the information is known. So we'll try to understand about the fibrous protein. Now this fibrous protein, they can be divided into two classes. Okay, so the first one is called as the keratin group. Okay, it is also called as the KMEF group. Now these are the initials of the amino acid sequence which could be present into it. Fine. So the keratin group is one of the what we can say class of the fibrous protein. So we'll talk about this keratin, what it is made up of, or how this particular protein is being made up. So it's a fibrous protein. So what the first thing should come to our mind, that it has to be linear, and if it is linear, so it will be either having a helical structure or it may be having an sheet structure. So I'm just bringing out the characteristic feature of these structures, and from that we'll try to understand that how this protein is folded and what are the amino acids which are involved into formation of that typical structure of the keratin. So in that, the first type of the keratin which you might have heard about, the alpha keratin. By any chance, can you tell me where do you find this alpha keratin? Yeah, I think I'm audible.
It is
Yes.
present in hair.
Yeah. So basically, it is present in the hairs. It could be present into the nails, the horns of the animals, okay, or the claws of the birds, etc., etc. So these are the different sources which you can say where this alpha keratin is present. So just imagine that how much strength it is giving. I mean, two extreme examples which I have quoted here. One is what you said is a hair, where hair it is very smooth. Okay, it could be flexible, not too much flexible or not too much of elasticity, but still it is a flexible. On the other hand, if we talk about the nails, horns, or the claws, so those it's a very strong type of structure. Fine. So such kind of structures are from this alpha keratin. So we'll talk about the characteristic feature. Now the question might have come to your mind that why it is called as an alpha keratin. It's a keratin protein, but why it is called as alpha? Because it is the protein which is dominated by the alpha helices. Okay, so it is called as an alpha keratin. Fine. Now maybe in this slide I'll talk about the theoretical points, and then in the next third or fourth slide I'll show you the images so it will be clear. So try to remember the sentences what I'm trying to speak here. So this keratin dominated by these alpha helices, or these alpha helices, they are the subunits. So they are made up of what? So these alpha helices subunits, they are composed of an central alpha rod domains of 311 to 314 residues. Okay, so it consists of an central rod. Okay, and this central rod could be made up of around 311 to 314 residues, amino acid residues. So these many residues, they are involved into the formation of the alpha helical structure. Now flanking this central rod, flanking म्हणजे काय? On both the side of the central rod. Okay, so flanking on this of the central rod, you have some residues, okay, which are present, but these residues, they are non-helical in nature, means they are not having any secondary structure. Either it could be helical structure or the beta sheet structure. It is simply an sequence of some amino acid residues. These flanking regions at both the side, that is the N side or the C side of the central rod, they could be vary with respect to their size and composition depending upon where that keratin is present. Okay. Now this central rod, it consists of four helical strands arranged as twisted pairs of two stranded coiled coils. See, you try to understand, try to read the sentence. What I'm trying to say here, that the central rod, it consists of four helical strands. I mean, there are four helices which are involved into it. Now, these four helices, they are twisted around each other in such a way that it is forming that four strand structure. Means you have four helices, keep them in front of you. So out of these four, two helices, they will get uh, bound to each other. Okay, so I told you about the ridges and the grooves. So just imagine that two helices, they have packed into each other. So you are getting that double helix, I can say here. Now that double helix will again get coiled onto the another double helix. And this is how you can say that it is forming that four helical stranded structure. Okay, so that central rod, it consists of these four helical strands which are arranged as twisted pairs of two stranded coiled coils. Now after studying that particular structure, they found that this central rod, it can be related to an alpha helix. Means it can be imagine similar to that of the alpha helical structure. See, I am talking about the twisted four helices. Okay, it is forming a typical alpha helix structure, but I am talking about this, that when it has formed that four helical twisted structure, even after that twisting, it is similar to that of the alpha helix. Only the difference which you can make out here, that the pitch is found to be 0.51 nanometer instead of 0.54 what we find into a single helical structure. Okay, so that was the structural feature what you can say. Now the next question comes, that how this central rod is being converted into an helical structure. What could be the sequence of the amino acid residues which is forming only the alpha helical structure? So after having the sequencing of that particular central rod, what they found, that this central rod, it consists of a repeating seven residue segment. So what they found, that there are seven residues which are repeated. Means 1 2 3 4 5 6 7, again 1 2 3 4 5 6 7. So like this there are repetitions. So you can say that those seven residue segment could be A, B, C, D, E, F, G, which is repeated n times. Okay, so that you have that 311 to 314 number of residues, and that particular sequence, or that many residues, they are forming the helical structure. Now what are these seven residues? So after studying the different type of the keratins in the different sources, from the different sources, what they found, although it's not a true repeat म्हणजे काय, असं फिक्स नाहीये की हे 1 2 3 4 5 6 7 तेच रिपीट होत आहेत, but some kind of similarity or some commonality what they found, that the A and D residue, लक्षात घ्या, the A and the D residue, they are usually non-polar amino acids. Okay, so it could be alanine, it could be isoleucine, it could be leucine, it could be valine. So A and D could be any one of this. On the other hand, the remaining residues, that is the B, C, F, they are polar in nature. Okay, and what are those? They are the aspartate, glutamate, lysine, arginine, alanine. Okay, so this gives us an idea, although there is no fixed amino acid which is forming this sequence. Okay, but it is the nature of the amino acid residue which is taken into consideration. So what they have found, that always at the first and the fourth position, that is A and D, you always have the non-polar residues, while the remaining region will be having the polar residues. Okay, now after telling this, and you people knowing the characteristic feature of the alpha helical structure, can you tell me what kind of helical structure will be formed here? Just guesses I want. याच्यामुळे तुम्हाला हेलिक्स कसा मिळणार आहे मला सांगू शकाल? येस, हे बघा तुम्हाला हेलिकल स्ट्रक्चरचे फीचर्स माहिती आहे, ठीक आहे. Now I have given you this information. So what do you think so? That what kind of helical structure you can get from this information? I don't know whether you understood the question or not. Fine. I'll present a whiteboard. This is just for your understanding. I'm trying to draw it here. See, that is why I ask you about these characteristic features of the alpha helical structure, where you said that it consists of a 3.6 amino acid residues, okay, in a given turn. Okay, so just imagine turn here. Okay, so maybe I will not be good at that particular drawing. So what eventually it will happen, suppose this was your first amino acid, somewhere here it could be your fourth amino acid, then fifth, sixth, and again the first amino acid will repeat here. Okay, so what it will happen, sequence specificity, that always at the first and the fourth residue you are having the non-polar residues, okay, while the other residues they are the polar in nature. Okay, so हे बघा, keratin, how many residues are repeating? 7. Now in a given turn, how many residues you have? 3.6. So it exactly half. 3.6 into 3.6, it comes to 7. So these 7 residues, they are forming 2 turns of the alpha helix. Now these 2 turns, they are formed in such a way that always the first and the fourth residue, it will be on one side, while the remaining residues will be on the other side. So here you can imagine that this side of the polypeptide, it is non-polar in nature. Okay, so due to this typical folding, whereby the first and the fourth amino acid residue, it comes on one particular side, so you may get an alpha helical structure which is amphiphilic in nature, means one side is non-polar in nature while the other side is hydrophilic in nature. I think you got this idea what I want to tell. Is it clear?
Yes, sir.
Okay, fine. So we'll continue. That is what is given in the next point only. So the non-polar residues makes the helix unstable. Okay, so the presence of so much amount of the non-polar amino acid residues may make that particular helical structure unstable, but this instability is immediately resolved by the association of the hydrophobic sides of the two coiled coil. So just imagine two coils, the hydrophobic end and the hydrophilic end. So although the presence of these non-polar residues is making it unstable, but what will happen? The hydrophobic regions of both these strands, they will come together. They will associate with each other. The grooves and the ridges, they will get buried into each other, and they will be forming an coiled structure. Okay, and that hydrophobic interaction between those two coils is giving the stability to the alpha helical structure. Right, so like this you may get the coiled structures which are formed. So that is what I have written here in brackets. See here, so 6 residues per turn, that is 7 आपल्याकडे आहेत. So 7 is equal to 2 into 3.6. So it aligns the first and the fourth non-polar on side to non-polar strip. Okay, so the two non-polar face of the helices, they associate to form the coiled coil. So these helices, they sacrifice the stability due to the twisted conformation, but again gain the stabilization energy from packing of the side chains of the helices. See what I told you, that this presence of this non-polar, which are bulkier in nature, so they may lose the stability, but they are gaining that stability again by packing these side chains into the ridges or the grooves of each other. Okay, so that is giving them stabilization energy, so that the structure which is being formed, it is a stable structure. So overall, when you're talking about the alpha keratin which is present into the hair, the alpha keratin, it consists of what you can say, a microfibril which is of 80 angstrom, and this microfibril is made up of protofibrils. So there are around 9 plus 2 protofibrils which are joined to each other, okay, which are associated with each other, and they are forming a microfibril. And uh, these protofibrils could be made up of 2 to 3 helical structures which are entwined to form the coil structure. Fine. So just imagine that you have one thread. सुतळी बघा, सुतळी ती पण एक ट्विस्टेड असते. ठीक आहे, तुम्ही ते एक सुतळीचा एक तुकडा घेतला, त्याच्याबरोबर तुम्ही अजून दुसरा तुकडा त्याला जोडला, तिसरा जोडला, असे तुम्ही 9+2 जोडले. सो यू आर गेटिंग अ व्हेरी स्ट्रॉंग रोप which is being formed. Okay, so from a thread you are getting a rope. The same way, from a helical structure you are forming that coiled structure, and like this many coiled structures which are associated, you are getting a very strong type of an rope like structure for the protein, and that is nothing but the keratin structure. Fine, so I'll show you the images so it will be clear to you. Yeah, so just remember the sentences which I told you. So this is the comparison between keratin type 1 and type 2. So what I told you, that it consists of a central rod. So the blue color what you see here is a central rod. Fine. So it could be made up of 3112, 3114 residues. Fine. While the two regions which are shown in this orange color, what you see, the light orange color. So these are the flanking regions. So N terminal domain and the C terminal domain. So this N terminal and C terminal, they are the non-helical structure without any defined structure. Okay, so those particular regions are there, and these regions, they can vary with respect to their sequence or respect to the size. So you can see here the dotted line. So in some cases they may be present, in some cases they may not be present, or their number will vary, their sequence may vary. But this blue region could be the conserved type of the region. Okay, while this flanking regions could vary with respect to the type and the number of the residue. Now this is how the coiling takes place. So you have a single alpha helix. This single alpha helix, it will form the coiled structure. So you can see here that these two helices, they have coiled around each other. So you are getting this coiled structure. Now this coiled structure will again get coiled with the another coiled structure. So you are getting this protofilament. Okay, so it is a pair of coiled coils. So you can see here two helices. Now they are coiled to each other, and then this protofilament, they will be forming the filament. And in the filament, what do you have? You have 4 right-handed twisted protofibrils. म्हणजे हे जे चार एकमेकांना वाउंड झालेले, तर ते हे जे स्ट्रक्चर फॉर्म होणार आहे, they will be again getting wound or twisted around the another 3 different types of the pair of the coiled coils, and they are forming this structure that is called as an filament. Fine. Although these helices, they are the right-handed helices, but they are twisted around each other in the left-handed. Okay, फक्त हा एक लक्षात ठेवा त्याच्यात, that these helices, they are the right-handed helices, but they are twisted around each other in a left-handed manner. whereby you are getting this coiled structure or the filament structure which is obtained. So the fibril which is present into the hairs, the keratin protein what you might have studied, so it will look something like this. Okay, so you can see here these are the 9 protofilaments, and inside you have these two protofilaments. So this is a 9 plus 2 structure of the microfibril which is present into the hairs. Okay, and this structure is giving that much strength to the hairs. Okay, so even if you try to pull the hairs with your two hands, you'll not be able to easily break it, because that strength is given by this coiled structure of the 9 plus 2 pattern. Now you might have seen that the hairs may have some amount of elasticity. You can stretch it, and that elasticity could be due to the alpha helical nature of the polypeptides which are present into it. Fine. So is it clear about this? The structure, how it is formed? Again, the small story which I told you, that if it is a single stick, you can easily break it. But if you have many that may, many sticks, okay, so it becomes a very strong type of an stick. You will not be able to break it. So this is what you can imagine here. Now another question comes. Now it is made up of helices, and we have seen that the helix structure is basically stabilized by the intramolecular hydrogen bonding. So within the polypeptide, you have the hydrogen bonds which are formed, that is called as the intramolecular. So that intramolecular hydrogen bonds, they are giving the stability to your alpha helical structure. Okay, so you can now say that these fibrils which are present into the hairs, they are stabilized by the hydrogen bond. Fine. Now the next question comes, that this keratin is present into the hairs. Now when you are washing the hairs, okay, so if it is dominated by the alpha helices, sorry, dominated by the hydrogen bonds, what should happen here? That your hair should get dissolved into the water. If you are taking a bath, you are washing your hairs, so the keratin should get dissolved into that particular water, but the thing is not happening like that. Why? Because these keratins, okay, along with the hydrogen bonds for its stabilization, they are also having the disulfide bonds. Okay, so these keratin, they are also stabilized by the disulfide bonds, and that disulfide bond formation gives the insoluble nature to your keratin, and that is why your hairs, they do not get solubilized into the water when you go for the washing. Fine. So two forces, what you can imagine, the first one is the hydrogen bond, and the second one is the disulfide bond, which is imparting the stability and the insolubility to your protein. Fine. Now knowing this particular background, you might have experienced, because many of us have straight hairs, many of us have curly hairs, or maybe if you are sleeping, if you are having a good sleep in the night, and when you wake up in the morning, you see that your hairs are standing. Okay, so you may look something like cruising or something like that. So to avoid that, what do we do? We simply take the water, apply it to the hair, and when you apply the water, what will happen? The hairs will again settle down. Fine. Or maybe you might have seen that some of them, they try to make their straight hair into a curly hairs, or some of them, they may try to convert their curly hairs into the straight hairs. That could be a thing. Or nowadays the fashion is there, in specifically in the boys, that they use some gel, and by using that gel they can have the different shapes to their hair, etc, etc. So why is it possible to change the nature, not nature, but the shape of your hairs? Why it can be done? Because the simple reason is that as I told you that it is having, or it is dominated by the hydrogen bonds, and it is also having the disulfide bonds. So it is very much essential to sorry, it is very much easy to reorient these hydrogen bonds or the disulfide bonds, so that you can have the different type of the shapes to your hair. So when you are applying the gel for setting your hair, just for curiosity, after the lecture you can see that what are the chemicals which are present into that. So in that you may find that there are some reducing agents. Okay, so when you are applying the chemical, what is happening, that reducing agent, it will be breaking your disulfide bonds. Okay, your means the hair's disulfide bond, it will break the disulfide bond. Now, although these disulfide bonds are broken, but they have a very high affinity for each other. So you can imagine that it is breaking a disulfide bond between two residues. Okay, but immediately these residues, they may form another disulfide bond, some other residue which is present. Okay, so there could be a mismatch type of a thing which will take place. Okay, so it will be simply an exchange of the disulfide bond from with one histidine form with one cysteine to the another cysteine. So due to this change into the disulfide bonding, you may have the different shapes of the what you can say hairs. Okay, or if you are using that you use that rods where heat is given to the hair, so that your curly hairs can be converted into the straight hairs. So if you are heating it, means you are providing the energy, and that energy will again break the hydrogen, sorry, the disulfide bonds, and that disulfide bonds may change their nature, so the curly hairs can be converted into the straight hairs. Or the other example, that simply after waking in the morning, you apply your water to the hairs, what is happening? Again, there is a change into the nature of the hydrogen bonds, and that is why your hairs can be easily set. Okay, so like this there are few factors which are important to know the structure of the protein, and that is what I have brought in here. So some forms of the keratin, they have the disulfide bonds between adjacent molecules, making the structure rigid, inextensible, and insoluble. So where are they present? In the claws, the fingernails, hairs, and the horns. So if you talk about the hairs, you may have the permanent wave. So permanent wave can be obtained whereby the disulfides are reduced and cleaved, then reorganized and reoxidized to change the degree of curl or the wave. Fine. So the way the thing what I was trying to explain you, that there could be a change in the partners for formation of the disulfide bond, and that change in the partner may lead to the curling or the wavy nature to your hairs, and that is why you get the permanent waves into that. Right. The same way setting, when you set the hairs, that is the setting hair or the curlers, they represents the rearrangement of the hydrogen bond between the helices and the fibers which are present into the hairs. हे बघा hairs मध्ये फक्त keratin नसतं बरं, it's only a protein, but along with that protein you have many fibers which are present into it, and then a hair structure is obtained. Okay, so I think the structure will help you to understand about a fibrous protein. So we have seen one type of the fibrous protein, that is the keratin. Specifically, we have talked about the alpha keratin, where it is linear and it is dominated by the alpha helical structure. So when you are talking about a keratin protein, the folding of this protein, it ends up at the secondary structure, that is the formation of the alpha helical structure. Okay, any questions up to this point? Yeah, have you understood what I'm trying to say or trying to explain here?
Yes, sir.
Any doubts, any questions, anything you want me to repeat? Okay, fine. It's a very small point, so I'll continue with this. I'll end up with this keratin part. Yeah, the second type of the keratin of the fibrous protein. So that second type is called as an beta keratin. Now, why it could be called as a beta keratin? Obviously, the logic will be that it is dominated by the sheets. Okay, so all those keratins which are made up of the sheet like structures, they are called as the beta keratin, or they are called as the fibrion. Any idea where this beta keratin will be present? Any guesses?
Carrot.
Yeah,
Carrot.
not able to get your voices. Okay, fine. So it is found in silk. One of the sources that, so it is composed of stacked anti-parallel beta sheets. Okay, so it is dominated by the sheets, and in that it is dominated by the anti-parallel sheets which are stacked one on each other. So this beta keratin, it is a large stretch of polypeptide sequence which is also made up of pseudo 6 residue repeat. Okay, pseudo it is called as, means you are not fixed that which residues are going to repeat, but it consists of an 6 residues which are repeated, and within that 6 residue every other residue is a glycine, means a glycine is going to be present into that particular 6 residues which are repeating. So you can say that the sequence could be glycine, serine, glycine, alanine, glycine, alanine. So you can see that there are many glycines every residue, म्हणजे एक सोडून एक always you will be having a glycine, आणि जो सोडून एक जो आहे, so in that region you may either have the serine or you may have the alanine. Okay, so these 3 residues which are continuously repeated, they form a 6 residue, and these 6 residues are then repeating in that particular manner. Now this beta sheet, it extends. Okay, the polypeptide chain which is formed, it is forming a sheet like structure. It is a extended structure, and it is extended in such a way that it will be having two sides, the above plane and the below plane. So what it has been found that due to this anti-parallel nature and the extended structure, what is happening that all the glycines, the R groups of all the glycines, they will be directed at one side, while the remaining residue, that is the alanine or the serine, they will be present onto the another side. Okay, so just for example, I can say that the suppose the glycine, they are directed towards the below the plane, while serine and alanine, they will be above the plane. Okay, so if I'm having a turn, means I'm reorienting the direction of that polypeptide, so I'll get an anti-parallel. आता तो चेंज केल्यावर काय होईल बरं सांगा? Maybe I'll go back to the what I can say. Whiteboard कुठे गेलं? Yeah, say suppose this is my polypeptide chain, fine. So I have the glycine on this side, so all the glycines will be on this side, and you may have a serine on this side. Okay, serine, alanine. Okay, so now this region, it has formed an beta sheet, but now there were some residues which we did not allow the beta sheet, so it caused an turn here. हे बघा आता हा टर्न होतोय. Now due to this turning, what has happened? It has changed its direction. आता ही आतली बाजू बघा. So this side you have the glycine. So here also you can imagine that there is glycine. So automatically you can see here that there is a glycine. Fine. So this due to this turn. Now again this side it will be what? Serine. इथं मी परत दाखवतो alanine. Now again there will be a turn. See here the turn. आता ही आतली बाजू ही बघा, ही if it is continuing like this, so what you can have? So again you can have a serine here. So see the nature that when it is folded, always you will have the glycines on one side, or the glycine of the two strand, they are interacting with each other, or the serine of the two strand, they are interacting with each other. Right? So such kind of nature is formed when you are having this particular repeats. uh, yes. So the pairs of this beta sheet, they pack together. And how they are packed? So glycine to glycine, or alanine to serine, or alanine to serine. So this is how the packing will take place. So I have explained you with the diagram. Now this keratin, it could also be found into the feathers of the bird. Okay, so that is also one of the examples. So here with this diagram, it will be easier to understand. So you can imagine that this is the extended structure of the what you can say, the beta keratin. So you can see this orange color, it is an alanine. Okay, alanine, and on the other side you can see here that there is glycine, fine. And these are the two anti-parallel. So all obviously this alanine and alanine, they will be on one side. And if you imagine here downside, if there is one more strand, you can say that the glycine and glycine will be on one side, fine. So this is how the fibrion or the beta keratin structure is formed, and it is dominated by the beta sheets. So that was all about the keratin protein, what we have talked about. Any doubts into this keratin protein? So tomorrow we'll take one more example of this, that is the collagen. Okay, and that will help you to understand about the fibrous proteins. And once we are done with it, then we'll talk few things about the collagen. Any difficulty? Any questions to be raised? Yes. Meanwhile, write your roll numbers in the chat box. Vaishnavi, write the numbers and send me across.
Yes, yes, sir.
Any questions? Okay. Hopefully all of you are coming on Monday. What's the status?
Yes, sir.
So all are coming or only few? All of you got the hostels?
Yes, sir.
Because I'll have to plan whether to have it offline or online. Let's see. Uh, Probably we'll have it online only, but in the afternoon we can meet in the department. Fine. And then from Tuesday we'll decide where to go, online or offline only. Okay. That is one thing. Then the second, have you all filled up the exam form?
Yes,
Anyone
sir.
remaining?
Yes,
Yes,
sir.
sir.
No, there were some 3-4 students who has not filled. Are they in the lecture? Just check in your group if anyone is remaining. I think the 15th is the last date. After that, they will have to pay the late fee. So just remind them, if they have not filled the exam form, complete it by today or by Monday, so that the things can be worked properly. And also all of you, make sure that you have submitted your application form, the hard copy, because it has to be sent for eligibility section, etc. So kindly do those things. Okay. If she has completed with writing the roll numbers, we'll call it for a day. Good day, all of you.
Thank you,
Yes,
sir.
sir.
Yeah, welcome.